Hey, hi everyone. I hope you all are doing fine. So in this video, we will understand a concept in the RNA sequencing world that's called Strandedness. So you might have encountered, some of the confusing terms like FR, RF, the strandness, the library type like fr-firststrand, or it's a second strand, or ISF, ISR. So these terms are confusing because when I started, it was confusing to me as well. And if you are using some of the tools, like the tool itself will ask you what is the library type that you got. For example, in feature count, if you have been using, it will ask you to input -s, whether you put 0, 1, 2, like that. Similarly, HTSeq asked for stranded, Salmon will ask you for library type. So that's what like we have positive strand, negative strand, we have genes coming from. So which gene is present in positive strand, negative strand, what does that mean by, and how do you, like, you get R1 and R2, and from there how will you derive how to like whether that's belongs to stranded protocol or non-stranded protocol, and if yes, then first strand and second strand. So all the concepts I will try to clear in this video. Okay, so stay tuned towards the end. So now let's start with the very fundamental concepts. Like we have two strands, right? And this concept of two strand and four views. So that's what we will see. So DNA is double stranded. So we have two strand. One is, let's call the plus strand and the minus strand. So these are complementary to each other. So for example, let's take an example: ATGC, GT, This goes from 5' to 3'. So the complementary strand will be A will like opposite is T to A, G to C, like that, right? So we have two strand, but the same two strand you can view in a 4 different ways, right? So this is what the plus strand, like the same green one you see, which goes from 5' to 3' view of the plus strand. And same, if you just reverse it, you will see it's not reverse complement. You have to just reverse it. That means C belongs to here, A, T, G, C, like that, just reverse order. That means same strand. Now you are looking into 3' to 5' direction. So now that's what it is written, 3' to 5' view of the same positive strand. So now this is the negative strand, the purple one, and which is the complementary of the one. Okay, so now this you are viewing from 3' to 5' direction, right? So, and the same, if you just reverse it, you will see 5 to 3 of the negative strand. So that's what two strand, but you can see into 4 views. So now you might be wondering why 4 views needed, because when we do the matching, right, so we have R1 and R2, when we try to align with the strand, right, so we compare against any of these 4 views. So your read may map to this view or this view. So irrespective of whether it matches to this and this, you say that okay, this is matched with the plus strand. Now same goes to if your read matches to this or this, then you say that it is matching with the negative strand. So in reality, it exists in DNA. We see this thing, but views, right, like when we do the matching and alignment, it's better to look into 4 views, so it will be more easy to compare. So that you will see when I try to portray that, you can easily understand. Okay, so 2 strand, 4 views. So now next one. So next important concept. Now this is the transcription, right? So now let's understand how RNA polymerase transcribe our DNA to RNA. So now RNA polymerase has to decide, shall I go choose the plus strand or shall I choose the minus strand? So that's important. So that's why, depending on the gene, okay, whether a gene, when we say gene is in a positive strand, gene is a negative strand. So depending on the gene, the RNA polymerase will now either he will use the plus strand or the minus strand and consider that as a template. And this is very, very important point. RNA polymerase always move from 3' to 5' direction and then synthesize the mRNA. So that means if RNA polymerase choose, oh no, I will now choose the plus as a template. So whatever the mRNA we will get, that will match with the minus strand because complementary, we will see. And if it chooses the minus as a template, then we will get the plus strand mRNA. So we say that gene is on the plus strand. We'll try to clear this concept. So now let's consider this first case. So RNA polymerase now decide, hey, I will go with the negative strand as a template, and then I will move from 3' to 5' direction, and then I will create an mRNA. So whatever the mRNA by this approach, you will get, right, that will match with the plus strand. And that's why we say that since the mRNA is the mRNA what we get is matches with the plus strand sequence, we say that, hey, the gene is belonging to the plus strand, or the gene's product is matched with the plus strand. So let's take an example. So the same view, the green is the 5' to 3' plus view, and the cyan one is the same one, a plus strand, but it's a negative reverse 3' to 5', and this is the complementary strand, minus strand, minus strand, and it has 2 views. So this I will always keep so that you will see the 4 views, and we will see. Okay, so now let's say, as I says, RNA polymerase will now decide, hey, I will choose the negative strand. And always, as I said, it moves from 3' to 5', right? So that means RNA polymerase will choose this, purple one as a template, and it will move from here to here. So that's why whatever the mRNA we get, that will be complementary to this one. So now what mRNA we get now? T complementary A is complementary to U. Now because it's RNA, right? So instead of T, we will get U, C complement G, G complement C, and similarly CA, TG complement GUC. So now this mRNA what we got is a complementary of the purple one. So the mRNA produced the yellow matches with the positive strand. Now you see, you take this one, now you can compare against 4, which strand it is matching? AUGC, ATGC, and GTAC, here GUC. That means now you can say that the mRNA is matching with positive strand, and that I have given it green color so that it is matching with this view of the positive strand. So now the conclusion is, now RNA polymerase considered negative strand as the template, and it produced an mRNA that matched with the positive strand. So that's why we say that, hey, this gene is on the positive strand. That means the gene is expressed from the positive strand, and that means the RNA polymerase considers negative strand as a template. So this is how you say that gene is positive strand. That means gene's product, if it is matching with the plus strand, then you say that the gene is on the positive strand. That means gene is expressed from the positive strand. Now let's take as example of the second case. So now RNA polymerase now say that, hey, I will now choose the positive strand as a template. So that means he will produce the mRNA. The mRNA sequence will exactly match with the negative strand. So now that's why we say that, hey, this gene is on the negative strand, or the gene's product mRNA that's matched with the negative strand. Same example, but now, as I said, RNA polymerase will consider positive strand as a template. So here positive strand, and that to be RNA polymerase go from 3 to 5, right? 3 to 5 side, right? So it will choose this cyan color as a template, and it will go from here to here. So whatever the mRNA we will get, that will be the complementary to the cyan one. So that's what we see to opposite G, A, U, T, A, G, C, like that. So that means now the mRNA what you got is like GUA, CG, C. Now you just take this one and compare with this one. So which one it is matching? It is matching with this view. That means that's why I've given the orange color. That means this particular mRNA exactly matches with the negative strand. So now we say that, hey, the mRNA matched with the negative strand. That's why the gene is on the negative strand, or the gene is expressed from the negative strand. So now I hope this got clear. So final conclusion, if mRNA sequence matches with the plus strand, you say that gene is from plus. mRNA if matches with the negative strand, you say that gene is on the negative strand. Okay, so now let's understand strand-specific RNA protocol. So far we got the RNA. So now we know that RNA we got either from, if a gene is positive strand, we got an mRNA. If a gene is from negative strand, we got an mRNA. So now this is what you are actually doing the sequencing, right? So now you take the mRNA, you are doing the transcriptomics, right? So you are doing the sequencing. Now here you decide whether so what happens in transcriptome? So you get an mRNA, right? So that you take that mRNA, basically, because mRNA you cannot sequence, so you have to convert that into DNA. So first you convert into a first strand. The first, this is the complementary of mRNA. Let's take an example here. mRNA is this one. So you synthesize the first cDNA. So okay, then in the second step you synthesize the second strand cDNA. Okay, so now RNA is converted into DNA. So now we can take this and do the sequencing, right? So two-step process. First you synthesize the first cDNA strand, and then you synthesize the second cDNA strand. Now after this, the sequencing protocol will vary depending on your experiment. So if you choose that, hey, I do not want any strand specific RNA sequencing, then you don't do anything. You go with straight away sequencing. So that means some of the reads will come from this strand, some of the reads will come from this strand. So you will get a mixed pool of both reads represented from the first and second strand. So this is the unstranded RNA sequencing protocol, you would say. But you say that no, I want to do the sequence the only one strand only. So here you have two options. Either you want to keep this one or you want to keep this one. Okay, so in this green one, you say that here you are considering you are deleting the second strand. Okay, so the second strand you are deleting it and you are keeping the first strand only. So that means all the reads what we get will coming from the this first cDNA is a template, right? So that will be template. But in the here alternately we are what we are doing, we are just doing the opposite one. Here we are deleting the first strand. Now we are considering second strand, yeah, so we are going with that. So that's what the name will be, fr-firststrand and fr-secondstrand. So if the library type or sequencing library type is fr-firststrand, that means you are keeping the first strand and deleting the second strand. This is the most common approach. Like for example, the Illumina, TrueSeq and other approaches are. And generally, this method is a dUTP method. That means while synthesizing the second cDNA, instead of D, so you add dUTP. Okay, so dUTP is incorporated during second strand synthesis, and then once is there, then there is some enzymatic digestion is done, and that will degrade the second strand. So that means your first strand will survive. I hope this is clear. So this is very important for the next, because we will looking into if you are going with first strand, then how it happens, and if you are going with second strand, how the this thing moves. That I will explain. Okay. So now if first cDNA sequence is retained or survived, so we call that it is first strand. If second cDNA sequence is retained and proceeded further downstream, we say that fr-second strand. Okay. So now let's see how. So you got your experiment is done. So now you decided which protocol, whether you want to go with this, first or second. Now you did the sequencing, you got R1 and R2, you got the reads. So now from R1 and R2, now how will we decide whether library type is first and second? Okay, so let's see that. So we'll first focus on the first strand protocol. That means that's where we are retaining the first strand. We have deleted the second strand, and again we'll go with the, let's say gene is in the positive strand. So what will happen? So if gene is positive strand, and from the gene we'll get mRNA, and the mRNA we convert into cDNA. We deleted the second DNA, and now first cDNA we take and we do the sequencing. We get R1 and R2. We take this R1 R2, we'll match it, and we will see how it looks like. Okay. So now the same thing. So again we have four views: plus, plus, minus, minus. This is the 5' to 3', 3' to 5', 5' to 3', 3' to 5'. Okay. So now, as I said, the gene is on the plus strand, and that means RNA polymerase will consider the negative strand as a template, and it will go from 3' to 3'. So it will go like this, and it will create the first mRNA. So this is the negative strand as a template, TACG, right? So TACG, this one same as the fourth line is here. So now RNA you synthesized is the complementary of this. So T becomes A becomes U, like that. So now this is what you got the RNA, right? This is the RNA what you got. So now the first DNA will be complemented to this, right? So let TACG, and again the second is deleted, but now the same you can view in the alternate form into 5' to 3'. That means this is the first cDNA. If you view from 5' to 3' direction, you will get this sequence. This is just reverse of CTA, right? The reverse of this one is GTAC, GCAT, like, okay. So these two are the reverse of each other. So as I said, to matching it would be very easy. So now you consider this first strand as template. So that's why this one, because 5' to 3' direction you have to keep, like 5' to 3' of the cDNA sequence looks like this. So now when you do the sequencing, what the sequencing will do, the read one will be exactly from this direction. So it will be GTACG, while the read 2 will be the reverse complement of this side. So that's what you will get the R2S. Reverse means T complementary is A, then A complementary is T, C complementary is G, and G is complementary C. So you get R1, and R2 is the reverse complement of this side. So now this is what the R1 and R2 you got. So now you have to take this one and you have to match with this where it is matching. So now you can take this R1, what you see, GTACG, where it is matching, GTACG. So R1 is now matching with which strand? Minus strand. So you say that now R1 matches with the negative strand, and you take R2, ATGCG, right? So this is matching with the plus strand. So now your rule what you got is like R1 is minus strand and R2 is positive strand, and your gene is on the plus strand. So now we create a rule like, hey, R1 is, so when gene is on the plus strand, so gene is on the plus strand, so we said that R1 is matching with the negative strand and R2 matching with the positive strand. So just opposite. Okay, so minus and plus. Okay, so the notation how you denote is like, because it's a R1, right, you say 1, and the which read, whether it's R1 or R2, then you say read is in which strand. Okay, and then gene is in which strand. This is how we unit, because we have two information, right? We have the gene information, the source, and the output. That means the input is the gene and output is the read. So we have to connect which strand to which strand. So in this, the first row, you see our gene was on the plus strand, and R1 matched with the negative strand. So we write the rule: 1 read was in the minus, but the gene was in the plus. So 1 minus plus, whereas 2, the second read matched with the same strand as the gene. So we get 2 plus plus. Okay, so you will see that this will be ultimately the first stranded the rules. Okay, so now let's take an example. Minus, that's why I have kept it blank, means we have not yet analyzed that. So let's take an example what will happen on the when the gene is in the minus strand. Okay, so we have same views, but now since the gene is on the minus strand, RNA polymerase will consider the plus strand as a template, and it will go from 3' to 5'. So here, in this case, it considered the negative strand as a view here. The fourth one it chose, but in this case, it will choose the second here, and it will go like that. So that's what the template what we choose is the second row. Okay, CATGC through, and you do the reverse. So now RNA will be synthesized, so it will be complementary. C becomes Z, A becomes U, T becomes A, like that. And the RNA what we got will match to GUA, CGAU. Now you synthesize the first DNA, so this will be the complementary, and you just take the opposite view as well. That means same first DNA, but we are referring into two views. So, and you keep the 5' to 3' view, and now you get the R1 here and R2 is the reverse complement side. So when you do this one, so what R1 we get? R1 is ATGCG, whereas R2 is the reverse complement from this side. So C becomes G, A becomes T, T becomes A, and G becomes C, and C becomes G. So now this is what R1, R2 got. So you take this one and you match where it is going. So now you say that R1 ATGCG, ATGCG, that means now R1 matched with the positive strand, whereas R2 GTACG, GTACG, so you are matching with this view particularly. So you say that now R2 matches with the negative view. That's why it is very easy to compare, right? Because if you have 4 views, it is easy to directly compare GTACG. Otherwise, you will be confused to do the reverse complement, try to get wonder. So that's why 2 strand 4 views is a better way to understand this concept, and that's what I thought to represent that way. So now the rule is like R1 is matching with the plus strand. So we feel R1 is matching with the plus strand and R2 is matching with the negative strand, and our gene was on the negative strand. So the rule what we got is now, okay, so 1 is the first read, and when it was in the positive strand, the gene was in the negative strand, and where for the second read, the gene was on negative and R2 matched with the negative strand. So now the final combined root is like 1 minus plus 1 plus minus. That means when read 1 is in matching with minus, you get plus gene, and same plus and minus, the opposite. But in case of second read, it will be plus plus and minus minus, means the second read will always match with the gene, positive gene or negative, like that. So if such kind of thing is observed, that means you took the R1 and tried to match with whether R1 or R2. For R1 it will be opposite, it will not match directly, okay, and for second it will match with the same strand notation, and this type of library is called the fr-firststrand library, where you are retaining the first cDNA and you have deleted the second strand cDNA and you removed. And the strandedness is generally you are using this term R and L, because okay, so the first read will be opposite, you see, minus plus plus minus, that means reverse, and the second one will be plus plus minus minus, means you are giving F and R, so like that. Okay, so now I know, just to revise it, it might be confusing, but yeah, this is how I could explain. So now we just saw first strand. Now let's say you have done the experiment. Now you are saying that, hey, I don't want to go with the first strand, I want to go with the second strand. So generally this method is generally called the ligation method. This is the earlier was the dUTP method. This method is ligation method, and basically this is from solid time. This is rarely used these days, but yeah, this is one of the method where you have the option that you delete the first strand and you go to the second strand. So again, we'll start the same approach. We'll take the positive and negative and start. Same view. the let's say gene is on the plus strand, so RNA polymerase will consider as a negative strand as a this one, and you sequence like this and you get an mRNA and use DNA. This is what you got, the RNA. So now the first strand cDNA will be like this, and the same first strand cDNA in the opposite view. And now you synthesize the second strand, and second strand 5' to 3' view. So all possible views I have kept, first and second strand. But now what we need to select, the second strand as a template, and always you consider 5' to 3'. And now you do the R1 this way and R2 this one. So R1 sequence what you get will be ATGCG like this, and while R2 will be the reverse complement, or like the C becomes G, A becomes T, T becomes A, like that. So finally this is what we get. So now you take this and try to match where it is going. So you say that R1 ATGCG, it is matching with positive strand. That means now R1 matched with positive strand, R2 matched with the GTACG, it will match with the negative strand. So this is what you say. That's when the gene was on positive strand, this happened. R1 matched with positive, R2 matched with the negative. So this is what the third rule you will get. Gene was on the positive strand, but here R1 matched with the positive. Okay, whereas R2 matched with the negative. That means here the read one is going same as the gene, but in this case it was just going gene. Read one was opposite to the gene, but here in this example read one is going same as this one, whereas read 2 is the opposite. So that's why you got 1 plus plus here, and but 2 minus plus. That means when read is negative, you are getting positive in the gene strand. Okay, so now we'll go look the same example, minus case. Okay, so gene is now on the minus strand. So RNA polymerase will now consider plus strand as a template, and it will sequence from this to this direction, and you get mRNA, and again 5 views, and finally you take this as template. This one survives, and you take the R1 and R2 this side, and R1 will be ZTACG, and R2 will be the reverse complement of this side, and you get this one, this one. So when you take this one and try to align, so you say GTACG is matching with the negative strand. So now R1 is matching with the negative strand, whereas R2 is matched with the this positive strand. So what concludes that, hey, if a gene is on the negative strand, we observe that R1 is also matching with the negative strand, but R2 is matching with the positive strand. So the now rule got shifted. So when the gene was negative, we observed that R1 also matched with negative, but R2 matched with the positive. So here you are getting 1 minus minus and 2 plus minus. So the rule is like 1 is plus plus, minus minus means this the read matches with the gene strand. Here minus minus means when it is minus, that gene is also minus. But for the second read, it's just opposite. When the gene is minus, read is going to with the positive strand, means opposite strand, like that. So if such kind of thing you observe, then you say that this is the second strand protocol or second strand library. Okay, and so now you say that the first one is like F means forward, that means plus plus minus minus, that means same to same. So you say forward, but for second read you have to do the reverse, because when plus is there you get minus, when minus to plus. So that's why you say F and R. So for the second you say R. Okay, so that's what these two terminology might be confusing, but this is from this rule you can see what. So this is what all the tools decide. So they take the read and they check which of these 4 views they matching, whether it's matching with what and what, and then it check what the gene is from, and comparing the gene strand along with R1 and R2 matching, they derive what kind of library type it is. I hope this is clear. If not, try to revisit this one, and I will attach the slide as well so that you can watch on your own. But now the same thing, if it is a single end, right, we don't have R2. In this case, what will happen? Same table, you just remove R2 and R2. So you will get like your gene was plus minus plus minus. So in R1 can be minus plus plus minus. So here R1 is going against your gene, okay, opposite, right? So you get minus plus, and here when the gene was in minus, R1 becomes plus, and this, so in that case your rule will be minus plus plus minus. So again this is the first strand, and you say that this is RF, and the second same will plus plus minus minus means this is exactly matching. In this case you say that it is a second strand method or FR. So same concept, just you have to delete this R2 column, then you will get an idea. So now how you can determine, because it's very, so that's why there are some tools are available. They use the same logic to derive, and one of the tool is, so there are different tools I have listed here. One of the most widely used is from RCQC, this infer-experiment.py. And there is some other tool, like, you can go to GitHub, this one, check strandedness. How are we stranded here? I like this word at the end. Yeah, check strandedness. And there is also Salmon. If you're using Salmon has automatically, it will detect. So you can also specify library type. Anything you give, it will try to auto infer. And finally, it will give you in output format. So that way also you can get to know what type it is. Now the other three are mainly, it is not, you cannot detect it's more of kind of verification. Okay, so you have done the experiment, you know that, and you want to assure that from the data also this comes out as it is. So one is the Picard collect RNA sequence matrix. So that's why I have written like it's a more of verification method rather than derivation. You cannot know, but you can verify. So that's why what we can do, like, it takes strand specificity as an input, and so that's why you can run multiple times and with different settings, and finally it will give the report, and from the report you can easily know what percentage correct strand reads matrix. Okay, and alternately, like, you can use Qualimap. Again, it will report the 5' to 3' bias, and also strand specificity stats it will give, and you can use that to know what kind of library type you got. And this is better one, but it is very slow, and you have to manually visualize like the colors of R1 and R2, and based on that you can decide what kind of strand it is. Okay, so I'll just explain with one example, this infer-experiment.py. So this is the tool you can go. So this is the command you have to specify, like the BAM file and the bed file. And so let's say hypothetically you get an output like this, the fraction of the reads fail to determine, because you cannot determine. So let's say ignore them, but whatever you got, and now they check the same rule what I did at 1 plus plus 1. So there are two rules, right? So in this table you see there are two possible rules. Either it will match with this one or this one. That in this example, 1 means read 1 match with the gene plus plus minus minus, that's the second strand protocol, okay, or the first one. So in this case, if you get 50-50, that means you have both combination. That means this is a not strand specific, this is a unstranded, you can say. The second is like, let's say you get 90 like this. If you get this kind of, for example, here which rule matched, plus plus minus minus, that means 1 plus plus minus, that means it went with the second stranded method. Okay, and yeah, and again you can go with a single end as well. So this will match with plus plus minus minus, means this is the second strand method. Okay, so now this is the most important slide where we have shown the strandness comparison. So you will see that the dUTP method is the generally the first strand method, because here you are retaining the first strand and deleting the second strand. This is the most common one you will see, okay, in the experiment. And this is the second strand where you are retaining the second strand and deleting the first strand, and generally the ligation method. And third is the unstranded method. So if you are using Trinity, you can give this parameter RF for this and FR for this. And if you are using Kallisto, you can use again RF stranded and FR stranded. And if you're using Salmon, you give ISR, and here you give ISF. Okay, how I remember? Because R is the first letter, okay, and F is the first letter here. Okay, so just opposite one. And star we don't use anything, it auto derives. Again, feature count, here you have to specify 2, and this case, which is the common one, the Illumina one, and here you have to give the 1, because here you are deleting 1, just opposite. Okay, and StringTie, again RF and FR we give, and RSEM we give 0, 1.5 like that. So means yes or no, like that. Okay, and some other parameter I have listed it down, and this is what I have explained, like in the table 1 plus minus minus plus 2 plus plus 2 minus minus, which rule fits here, and the plus plus minus minus plus minus minus plus is the second strand rule fixed here. Okay, yeah, so there are some further resources that you can, I have listed it, where very nicely extra informations are there. So one is this blog, you can go. Okay, so this explains the same concept like what I have explained, and also it talks about further details like what is loss of strand specificity, and mainly how this information is encoded in the SAM tags like TS and XS. That I'll not be covering that, but you can read that. And second link is this is the one very comprehensive, like detailed. You can read Griffith Lab, so they have explained like which tools and what, and some extra examples they have given, and you can go through that. And this is the same one. And one more blog I found, like like that explains a little bit with the diagram what I have explained, but in a much better way as well. So you can just read that to understand. Okay, I hope this was very useful, and that's all I think I had. So in short, just let me give a quick summary. Like we started with different views, so 4 views. Okay, and RNA polymerase will always choose any strand, positive and negative, depending on the genes, and choose always go from 3' to 5'. That's the thing you have to remember, and based on that you can put where this arrow mark, and then you get the mRNA. And once you get the mRNA, you decide, you get the first strand, you get the second strand, you decide whether you want to retain this one or this one. Based on that, your protocol will change. And if you are using the first strand, so I think the final table I'll show. This is what the final thing, like so. And this is the final one. So if it is the first strand, so here you're retaining the cDNA. This is the UTP method, and that's where you will get this kind of observation where the read one will not match with the gene strand, whereas read 2 will match with the gene strand. And this protocol is the opposite route. Okay, and based on that, we also saw the single end, how it will look like. And we saw some of the tools that can be used to take the BAM file. It will automatically tell what strandness it is, and once you know how you can pass that information as per the different tools that you use. I hope this video was useful. So if yes, share it among your network, and yeah, I will maybe come up with some other videos. Thank you so much. Thanks for your time.